Hi, welcome to this week's problem solving session. I'll just be waiting for maybe 10 more minutes to see if there are other people joining and then I'll start. All right. Yes. Sunil, you had a doubt? You've raised your hand.
No, no.
All right. Sure. All right. Abhishek, you had a doubt? Abhishek, your hand is raised. Do you have a doubt? All right. All right. I think these are all the people that might be joining today. In case there are more, I'll let them join. I hope I'm audible to all of you. Can one of you just unmute yourself and tell me if I'm audible?
Yes, ma'am.
All right.
Are,
Thank you. All right. So I'll just give you a brief introduction. Yeah. Abhishek, do you have something to say?
My name is Abhishek Kumar.
Yes. Hello.
जी मैम.
अच्छा आपको कुछ बोलना है? कोई कोई क्वेश्चन था आपको?
नहीं मैम.
अच्छा. All right. Thank you. Okay, so I'll just give you a brief introduction of myself. My name is Anakha Nair, and I'm a PhD scholar at IIT Bombay, and I'll be taking your weekly problem-solving sessions for the course of Basics of Crop Breeding and Plant Biotechnology. So I'll also be recording these sessions that we're having, and I'll be posting them to YouTube. The link of which the NPTEL team will be giving to you in case you're not able to join us live for the problem solving session. You can later watch it on YouTube. All right. Um, so, for those of you who were not there for the last session, I'll just tell you what these problem solving sessions are about. So, each week you will have some modules, right? And I'll be taking problems that are in those modules and we'll be solving them together. They won't be the same ones that are there in your assignment. If you need help with your assignment or if you have doubts in your assignment, we can sort them together after the due date has passed for these assignments. All right. So these questions that I'm taking right now might not be there in your assignments at all. Okay. So if no one has any doubt, I'll move on to the first question. You can stop me anytime that you have a doubt and I'll explain it to you again. Not a problem at all. All right. So please just feel free to stop me. So, and please answer the questions if you know what the answer is. All right. So just try guessing it. Even if you don't know it, whatever you think might be the right answer, just try guessing it. All right. So the first question is which is true for dominant gene transfer through backcross? So you must have learned about backcross method of gene transfer this week. So what they're asking is which is true for dominant gene transfer through backcross? And there are four options given to you. Option A is a recipient parent has dominant allele for the concerned trait. Option B is donor parent has dominant allele for the concerned trait. Option C is recurrent parent has to be used one time and Option D is donor parent has to be used several times. Out of these, one is correct. So, does anyone know what the correct answer is? You can either unmute yourself or write it in the chat box, anything that is comfortable to you.
मैम ऑप्शन ए हो सकता है क्या?
हो सकता है, देखते हैं। किसी और को है, किसी और के पास, does anyone else have a answer that they want to see? All right. No one else is sure about it. So, sure, let's see. So, what they're asking is which is true for a dominant gene transfer through backcrossing, right? And as you know, backcrossing is when you have a donor and a recipient, and the F1 progeny that you get, you keep uh, backcrossing with the recurrent parent, right? So, just so that it's clear, let's try this once. All right. So what they're asking you is which is true for dominant gene transfer through backcross. So your donor in this case, right, and the recipient. Okay. So the donor will be in this case the one who has the dominant trait. In this case, let's say AA. Okay. And the recipient, or in this case we also call them as recurrent parent, right? Because this is what you will be back crossing them with again and again, will be recessive. Okay. Because in this case, what you want from the donor, the recipient or the recurrent parent has everything else that you require, the yield, everything else. This is what you want your final plant to be like. The only problem with them is maybe something like they are not drought resistant, and you want to make them drought resistant. Okay. And that gene is present in the donor, and that is a dominant gene. So that means you need two dominant alleles, capital A, and only then the drought resistance will be shown. Okay. So you cross these two and you get F1, which will be capital A, small a, right? And in this case, this is drought resistance. Drought resistance is an example that I'm taking, obviously. Okay. And since this is a dominant trait, this will also be drought resistant. All right. This is not drought resistant. Just one sec. Someone else is joining. All right. So this is not drought resistant. Okay. And this F1 progeny that you've gotten, you will cross with the this recurrent parent again, that is small a. Okay. And this will keep continuing onwards, okay, till you get the final plant which has almost all of the phenotypical characteristics of the recurrent parent. Okay. And the drought resistance, that is capital A genes. All right. So that is your end goal. So the answer to the first question is option B, that is the donor parent has the dominant allele for the concerned trait. The recipient parent does not have the dominant allele like we saw. It has all the other phenotypical characters that we want. The recurrent parent has to be used only one time. That is also not correct. It has to be used multiple times. And the donor parent has to be used several times is also not correct. The donor parent has to be used only one time. And that is when you're crossing the recipient parent and the donor parent to get the F1 progeny. Was this clear? So the answer was not option A. Okay. The answer is option B. Does anyone have a doubt with this question?
यस मैम, ये रिकरेंट पेरेंट क्या है मैम?
रिकरेंट पेरेंट वो होता है जिसके साथ आप फिर से जैसे इसमें था कि हाँ आपका डोनर था ए कैपिटल ए, आपका रिकरेंट पेरेंट या फिर रिसिपिएंट है स्मॉल ए, ठीक है। अब एक मिनट, समवन एल्स इज आल्सो ज्वाइनिंग। ऑलराइट, सो ये स्मॉल ए वाले हैं। आई एम जस्ट एक्सप्लेनिंग इन हिंदी फॉर दोज हु मे बी अंडरस्टैंड हिंदी। आई विल एक्सप्लेन इन इंग्लिश ऑल्सो, सो डोंट वरी। ये स्मॉल ए वाले रिसेसिव जीन्स वाले हैं, बट इनके पास बाकी सारे वो कैरेक्टरिस्टिक्स हैं जो आपको अपने प्लांट फाइनल प्लांट में चाहिए। तो इसमें हो सकता है बहुत सारा अनाज मिल रहा हो, ठीक है, एंड बहुत स्ट्रॉन्ग प्लांट हो, बहुत लंबा प्लांट हो, ये सब इसमें है। बस इसमें एक प्रॉब्लम है कि ये फॉर एग्जाम्पल ड्राउट रेजिस्टेंट नहीं है, और आपका डोनर जो है वो ड्राउट रेजिस्टेंट है, बिकॉज उनके पास ये कैपिटल ए जीन्स है। ये कैपिटल ए इज अ ड्राउट रेजिस्टेंट जीन, ठीक है। सो इन दोनों को क्रॉस करने में, मतलब इन दोनों को एक साथ फर्टिलाइज करने में, आपको जो प्रोजेनी मिलेगी, ऑफस्प्रिंग मिलेगा, उसका जीनोटाइप होगा कैपिटल ए स्मॉल ए, ठीक है ना। बिकॉज इसमें दो जीन्स, दो एलील्स बनेंगे, स्मॉल ए कैपिटल ए, ठीक है। अगर आप एक पुनेट स्क्वायर बनाओगे इनका, तो आपको पता चलेगा कि सारे F1 प्रोजेनी का जीनोटाइप होगा कैपिटल ए स्मॉल ए, ओके। तो ये आपका F1 प्रोजेनी हुआ। अब इसको आप क्रॉस करोगे इससे, स्मॉल ए से, ठीक है। बिकॉज आपका एंड गोल ये है कि आपको सारे कैरेक्टरिस्टिक्स स्मॉल ए के चाहिए, जस्ट उसमें कैपिटल ए जीन्स भी होने चाहिए। आपको इस प्लांट का और कुछ नहीं चाहिए, डोनर प्लांट का और कुछ नहीं चाहिए। तो आप हर स्टेज पे बार बार जो भी आपको प्रोजेनी मिल रहा हो उसको इस रिकरेंट पेरेंट के साथ क्रॉस करते जाओगे, ताकि आपको एंड में ऐसा प्लांट मिले जिसकी सारे प्रॉपर्टीज इसके जैसे हो, बस उसमें ही कैपिटल ए जीन। इस बार समझ आया?
ओके मैम, ठीक।
हाँ,
यस मैम,
ऑल
समझ
राइट,
में आ गया।
ऑल राइट। एनीवन एल्स हैज अ डाउट? All right, so I'll assume there is no more doubt, and I'll move on to the next question. So the answer for this question was that the donor parent has a dominant allele for the concerned trait. Okay, so question number 2 is, if the dominant allele of a gene produces a anti-nutritional factor for generation of a crop variety through back cross breeding method, which of the following procedure has to be followed? And there are four options given to you. Option A is dominant gene transfer, option B is recessive gene transfer, option C is dominant as well as recessive gene transfer, and D is recurrent selection. So, do you guys have a guess of what the answer can be? Just you guys can just genuinely guess. There's no problem with that. You can guess what you think the answer is, and you can write it in the chat box. So I'll wait for maybe a few more seconds to see if.
Recessive gene transfer, B.
All right. So recessive gene transfer is one of the options. Anyone else has a different answer?
Ma'am, option B.
Option B. Okay. So mostly everyone thinks it's a recessive gene transfer. Does anyone think otherwise? कि कुछ और हो सकता है? All right. That's great. The answer is in fact B. It is recessive gene transfer. So for people who might still be confused, so the question is, if the dominant allele, that is in this case, let's say AA, produces something that is anti-nutritional, so that means that's not good for your plant, right? For generation of a crop variety through back cross breeding, which of the following procedure has to be followed? So you don't want this gene because it's anti-nutritional. Okay, you want the small a version, and that is the recessive gene. So you need to do a recessive gene transfer. All right, and recessive gene transfer is also like what you saw in the dominant one, except what the donor will be the recessive one. This one will be the donor. All right, and this will be the recurrent parent. The dominant allele will be the recurrent parent, and the small p, sorry, I don't think the difference here. Small p will be the donor. So you will cross these and you will get a capital P, a small p. That is the F1 generation, right? And this you will cross with again capital P. That is the dominant trait, right? And in the following series, what happens is you will either select or you will self these plants, and at some point again you will cross it with the recurrent parent, which will be this capital P, capital P. पिछले वाले question में it was the opposite, where the recessive parent was the recurrent parent, right? And यहाँ पर the dominant parent is the recurrent parent, because the gene you want is this one, and the gene you want is the donor parent. All right, so the answer, you guys were right, it is recessive gene transfer. All right, does anyone have a doubt with this? All right, so I'll assume no. If you're not comfortable unmuting yourself, you can obviously write it down in the chat and I'll be reading it. All right, so let's move on to the next question. So the question is, maintenance of a parental record are needed for which breeding method? And there are again four options given to you: pure line selection, backcross,
Pedigree method.
pedigree method. All right. So someone thinks the answer is pedigree method. So yeah. So option C was pedigree method and option D is bulk method. So anyone else thinks it's something else? We have discussed backcross breeding a lot, and pure line selection is when you want a pure line with the homozygous trait, right? And bulk method is where you want a homozygous plant, but in bulk you're doing it, right? And pedigree method is where
मैम ऑप्शन सी
option C, yes. Option
जी
C
मैम
is correct. So the answer is in fact option C. It's pedigree method. I was just explaining what is the difference between all four of them. So in pedigree method, you are keeping track of each cross that you're making, each selection that you're making, right? And each plant that you are choosing, you are keeping a track of it. So you maintain a very good parental record for pedigree method. Okay. So I'm assuming no one has a doubt in this. This was a more simpler question, right? So if everyone's okay with it, I'll move on to the next question. Just stop me in case you have a doubt. So the question number 4 is again, it's a simpler question. Johansson proposed pure line theory in which crop? So Johansson proposed the pure line theory. Which crop did he use for his experiments? There are four options: cereal crop, oilseed crop, conifer, and pulse crop.
Pulse crop.
All right, pulse crop. Does anyone think it's something else? Yes. So the answer is in fact pulse crop. That is legumes, right? And Johansson used princess seed, right? Uh, yeah, to propose his pure line theory, right? Uh, I think the scientific name is Phaseolus vulgaris, right? So that is what he used. So that is a pulse crop. So if no one has a doubt with this, I'll move on to the next question. So question number 5 is a pedigree record of 9214 hyphen 5 hyphen 4 F4 designates progeny of plant growing in F3 generation in row number 4. It was derived from the progeny of plant growing in F2 generation in row number 5. In which year the cross was made? And there are four options given to you: 1492, 1400, 1992, and 2092.
1992 C.
Yeah, so this is slightly a trick question. So if you remember from the uh, class,
मैम आंसर नंबर बी माने ऑप्शन बी
B तो नहीं. It is option number C ही है correct answer, because if you remember from the lectures, what they say is the first two digits indicate which year this has been in. Okay, and since pure line method, pedigree method was found in 1927, so anything before that is obviously not true, right? And it can be only after that. But since we haven't reached 2092, it can also not be the correct answer. So the only correct answer in this case is 1992. Is that clear? All right. So the correct answer is 1992, and you get that because you can see the first two digits is 92. All right. So if there are no doubts, I'll move on to question number 6. So question number 6 is choose the correct option for back cross breeding. And this has multiple answers from this. Okay, it can be one, it can be more than one. All right. So the four options given to you are: it is a practice that has been used by breeders for cross pollinating crops. Option B is back cross breeding is a new technique developed for uh, apomictic plants. Option C is back cross breeding is repeated until the offspring recovers about 99% genes from the recurrent parent. And D, back cross breeding is often used to increase the yield of the crop along with the rectification of a minor defect.
Option D.
Option D.
Rectification of minor defect.
All right. Anyone thinks it's something else? All right. So since they have mentioned this is a MSQ, it means it's not exactly, and there is a chance that it's not just one correct answer. So you were right partially. So one of the answers is option D, where the back cross breeding is often used to increase the yield of the crop along with rectification of a minor defect like resistance to some uh, beetle or bug or drought resistance or something like that. Okay. And the other correct one, that is, there are two answers, right? Is option C. So back cross breeding is repeated until the offspring recovers about 99% genes from the recurrent parent. So if this is the recurrent parent and this is the donor, you keep crossing the offsprings F1 and more with this recurrent parent till or every other phenotype that you can observe is the same as this parent except for this trait. Okay, so these two are correct, and the reason the first two are not correct is because it is practice used by breeders for self pollinating crops. Okay, so it's not, if this would have been correct if it was self, but it's written cross pollinated, so that's not correct. And it is a new technique developed for uh, apomictic plants. Apomictic plants are those which don't require fertilization, right? They like seeds from lemon, right? They don't require fertilization for forming seeds. Okay, and this is obviously not correct for that because you're not crossing anything in these kinds of plants. So these two are wrong. All right. Anyone has any doubt in this question? All right. So let's move on to the next question. Progenies divide from single self-pollinated homozygous plants are called as, there are four options: multiline, pure line, isogenic line, and inbred line.
Pure lines.
All right. Anyone else thinks it is something different? All right. So let's see the answer. The answer is in fact pure line. So pure line is progenies derived from a single self-pollinated homozygous plant. All right. Multi-lines are there are several pure lines which differ in only resistance to some particular thing. Okay, and they are produced by back crossing with the recurrent parent. Multi-lines use the same recurrent parent to give you different pure lines with different kinds of resistance. Okay, pure line is this exact thing. Pure progeny is derived from single self-pollinated homozygous plants. Isogenic lines are those which have the same genotype, okay, except for in one gene. Okay, isogenic lines have the same genotype except for in one gene where they'll be different. Okay, and inbred are cross pollinated species that are continuously inbred, that is, they are self-pollinated to get a homozygous line. Okay, so that is the difference between these, and the correct answer in this case is pure line. Does anyone have a doubt with any of the um, explanation for any of these lines or with this question itself? All right. So let's move on to the next question. Selection for simply inherited traits starting from which generation? Starts from which generation?
F2 generation.
All right. Anyone else thinks it's different? All right. So the reason for this, the answer is in fact F2, is because imagine this is your starting, right? And the F1 generation is capital A, small a. Okay. And if you remember from the Punnett square, right? You get all progenies will have the same phenotype and the same genotype, which is capital A, small a. Okay. And once you self this to F2 generation, you get capital A, capital A, capital A, and small a. So it is at the F2 generation that simple inherited traits can be started to select. Okay, like you can start selecting from the F2 generation, because F1 generation is the same phenotype and the same genotype. Okay, you won't see any difference in the characteristics in F1 generation. Okay, and you won't see anything different in the parents also, because they're homozygous. So only from F2 and beyond will you start seeing difference in traits. Okay, so the answer is F2. Anyone has a doubt in this question? All right. So let's move on to the next question. Question number 9 is the formation of extreme phenotypes observed in segregating population compared to the phenotype observed in parental line is called as isogenic line, transgressive segregants, pure line, heterobeltiosis. Yeah, I don't know how to pronounce this, but
B. Transgressive segregant.
all right. That is, in fact, the correct answer. It is transgressive segregants. So they end up having more yield or something better, extreme phenotypes than their parents itself. All right. So the answer is, in fact, transgressive aggregates. Isogenic line, like I had mentioned to you before, is where all the different plants have the same phenotype, same genotype, same phenotype, except for in one gene. Okay, so only one gene will be differing in them. Okay, pure line again is homozygous plants which have the same pure lines. The difference between pure lines and isogenic lines is that they are homozygous for one trait. So pure lines are homozygous for only one trait. Okay, isogenic lines are same in every other way except for one trait. One trait. Okay, and the last one is when hybrid performance is better than its best parent. Okay, and so you might be asking if this is also having a better performance than its best parent, and this is also having a better performance than its best parent, what is the difference between these two? Okay, so the difference is that transgressive segregants will show you better performance only after from and after F2 generation. But in this case, in heterobeltiosis, the better performance is something called hybrid vigor, where F1 will have the most amount of better performance than its parents. Okay, because it's a hybrid. So hybrids perform better here. Okay, so does anyone have a doubt in this question? All right, so let's move on to the next question. This is the last question for today. There are only 10 questions. So the question is, which method is mostly used for the transfer of cytoplasm?
Backcross method.
Again, yeah, that is the correct answer. So the answer is in fact backcross method. For those, like you would have seen in the class, right? So you have one nucleus and you have another one, and you want to transfer the cytoplasm in this to this. Okay, so what you do is you backcross. So this will be the recurrent parent in this case, and this will be the donor. Okay, you keep back crossing F1 and whatever generations you get with the recurrent parent again and again, so that you get the nucleus that is in the recurrent parent and the cytoplasm of the donor. So the answer for this is backcross method. Does anyone have a doubt in this question? All right, no. Does anyone have any other doubts? Because these are all the questions that are prepared for you.
Madam,
Huh?
in a study material, we are receiving only PowerPoint presentation.
Huh.
Can we have any notes about these chapters?
No, I don't think so. I think they will only give you PowerPoint presentations. If you have any doubts regarding this, you can write it in the announcements wala pages there, na? You
Yes.
can ask there. If the professor has the notes for this, he will provide it to you. But right now, it's only, I've seen in whatever week, it's only the PowerPoint that they're giving you.
Last week we have missed the meeting due to some reason. How we can get that material of discussion questions and all that?
Of this problem solving session.
Yes, yes.
Okay, so I have uploaded it as a YouTube link. Okay, I think the NPTEL team will be sharing the link with you, so you can see it. I'll maybe add it here also, so people who are there in the meet can see it. Just one sec. Okay, I hope you can see it in the chat, which is
Yes,
I think the
yes, we have
YouTube
received.
link. And I'll just also share my PPT. I have made a presentation folder, and you can see the PPT also in case you just want to go through it. This, I think the NPTEL team will also share with you, but just in case you need them right now. So don't worry
Yes.
if you're not, it's not possible since it's a Tuesday. It might not be possible for you to attend each class, but I'll be uploading similar YouTube links, and you can always go back and see them. Or if you have a doubt with any of the classes that you've missed, you can ask me in the next one. I might have said something wrong or something. If you have something like that that you think was wrong, just let me know in the next class. I'll make sure that I correct it. All right. Is there any other doubt that anyone else has? All right. So, if you guys don't have a doubt, you guys can leave the meeting. If you have any doubt regarding any of the modules or the questions that we've taken today or the one from last time, you can ask me. I'll still be in this call till I think about 9 o'clock. If in case there are other people who have not joined now, will join later. But if you guys don't have a doubt, you guys can leave. Hi, do you have a doubt?
Uh, just a minute.
Sure.
Yes, thank you, madam. We have received that link.
Yeah, sure.
Thank you.
Yeah.
Hello,
Yes,
hello,
yes, yes.
ma'am. I have go through that link. There is no any visible screen. Only first page is seeing on the YouTube.
YouTube में just
Yes,
one second.
only basics of crop breeding and plant biotechnology title is there, and your background voice is there, but there is no
Oh,
any screen. We
oh, I
cannot
did
see any
not.
text
Yeah,
in
thank
each
you for
slide.
yeah, thank you for letting me know. I'll have to re-record this. Yeah,
Yes. Okay.
yeah,
Thank
sure.
you.
Uh,
Otherwise, where you have shared a PPT?
PPT, I've given you a drive, right? There's one more link of the drive. I can pin this. It will be in the pinned comments if you see. There's a
Yes, yes,
folder
drive.
link. Huh.
That is also enough for us.
Huh. Okay. I'll anyways upload a new one. I didn't
Yes,
realize
sir.
nothing else had been captured. Yeah. Thank you for letting me know.
Yes, thank you, madam.
All right. Hi, were you in this meeting a little while before? Vivek? Am I audible to you? Vivek? Yeah, you can just write your responses in chat. You don't have to unmute yourself. Were you there in the session that I took a few minutes ago? Or this is you just joining now? Mm. Vivek, can you just write in the chat? Like you want me to solve the questions, right? Am I not seeing it? Am I not audible or is there some problem with my network? I can't hear Vivek. Uh, even though you are unmuted, I can't actually hear anything from you. Can you just write in the chat box? So I'll just start taking the questions. I just wanted to make sure that you weren't there in the first batch of it, in case you already have seen this question. So just stop me anytime, okay? So um, I think I'll start with introducing myself. Hi, I'm Anaka Nair, and I'm a PhD scholar at IIT Bombay. And I'll be taking the problem solving sessions for the course Basics of Crop Breeding and Plant Biotechnology. So I'll be taking problem solving sessions for week 2. Oh, okay. Yeah, so these questions are from the portions in the week that I'll be taking. So the questions I have today are from this week, but they are not the same ones that you see in your assignment. We'll be discussing the assignments if you need it after the due date is over. Okay. So can I start with the first question?
Yes, ma'am.
All right, sure. So you can just write the answer in the chat box if that is what you're comfortable with, not a problem, and just stop me anytime you have a doubt. All right. So question number one is which is true for dominant gene transfer through backcross? There are four options given to you, and they're talking about dominant gene transfer using backcross. Okay. So option A is recipient parent has dominant allele for the concerned trait. Option B is donor parent has dominant allele for the concerned trait. Option C is recurrent parent has to be used one time, and option D is donor parent has to be used several times. Can you guess what the answer is? All right. So let's just see how this works, right? So recurrent cross is where, let's assume something like this. This is the dominant parent, this is the recessive parent, right? And you cross them and you get a hybrid which is F1, okay, and they will show the dominant trait because they have one dominant allele, right? And you cross this with something called a recurrent parent, okay, and that is how all back crosses work, right? In the case where you want for dominant gene transfer, so what you want is to transfer the dominant gene to the plant of your interest. So we are going to assume that the plant of interest is recessive in the gene that you want to transfer to it, and you have a donor which has the dominant genes, and it's the donor, right? So you get the hybrid which is F1, and what you will do with this is just one second. Yeah. So you have this hybrid which is dominant for this trait AA, but it's not a homozygous um, dominant, right? It's a heterozygous dominant because it only has one capital A allele. Okay. So what you want is you want the characteristics of this plant in every other way, okay, except for this particular gene. So you want everything about this plant to remain the same except for this gene. Instead of this gene, you want it to become AA, capital A, capital A. Okay, you want the dominant traits. Everything else in the recipient plant or the recurrent parent is what you want to remain the same. So you are transferring into this plant this gene. And to do that, what you do is you have the F1, you cross it with this recurrent parent. Okay, and similarly, you get another offspring. Okay, so you will get AA and AA, right? So you will select this plant and then just one sec, there is one more person who's joining. All right. So again, you will cross it with the recurrent parent. All right. And you this keeps happening. So this is what a dominant gene transfer will look like. So the answer to the first question is option B. Okay. So the question is for those who joined just now, it is which is true for dominant gene transfer through backcross. Okay, and the correct answer is option B, where the donor parent has a dominant allele for the concerned trait. Option A is not correct because the recipient parent has a dominant allele for the concerned trait is not it. Since you only want to transfer the dominant gene, it has to be transferred into the recipient parent, okay, or the recurrent parent. And option C is not correct because recurrent parent has to be used multiple times and not just one. And option D is also not correct because the donor parent has to be used several times is also not correct. The donor parent has to be used only once. Okay. So if there is any doubt in this question, just ask me or write it in the chat box. If not, I'll move on to the next question. All right. So question number 2 is if the dominant allele of a gene produces an anti-nutrient, that is not good for your plant, you don't require this, right? For generation of a crop variety through backcross breeding, which of the following methods has to be followed? Option A is dominant gene transfer, option B is recessive gene transfer, option C is dominant as well as recessive gene transfer, and option D is recurrent selection. So, what do you think the answer is? Anyone? You can just write it in the chat. You don't have to unmute yourself in case you're uncomfortable with it. Just try guessing the answer. All right. I don't see any answers. But in the question, they're asking you that if there's a dominant allele, okay, and it produces something that is not good for your plant, that is an anti-nutrient, okay, how will you back cross it to get a plant that does not have this? So that means what you need is the recessive gene. You need a plant that is homozygous recessive, so that no matter how many times you cultivate it, you will keep getting a plant which is homozygous recessive and it does not have this anti-nutrient, right? So in that case, what happens is you cross them, you get the F1 which has this anti-nutrient still. Okay, so what you will do is, in this case, only this is what you require. Okay, everything else about the plant is, unlike in the last example in which it was the dominant plant's more phenotype that you required, that is, everything else about that plant required, and you needed the dominant genes from the donor, right? But this is not the case here. In this case, you want all the properties of the dominant plant, but you just don't want this particular dominant gene. Okay, you want the recessive genes. So this you will cross with this. So this is the recurrent parent and this is the donor. Okay, and this will keep happening. You will keep crossing the plants that you get with this recurrent parent. So in this case, the answer to this question is B, recessive gene transfer, because you're transferring this gene. Okay, this is the donor in this case. Okay, and this is the recurrent parent. So this is a recessive gene transfer. All right, someone had. Uh, does someone have a doubt? All right, I'm not seeing any doubts in the chat box, so I'll assume you guys have understood it, and I'll move on to the next question. So, maintenance of a parental record are needed for which of the breeding methods? Pure line selection, backcross breeding method, pedigree method, and bulk method. This is a very simple question. If you've paid attention in the videos, then you will know this, right? Yes. So, as Vivek is saying, the answer is in fact pedigree method. So, in pedigree method, you will need to have a very good parental record for each plant that you're selecting, right? Yeah, Satya, you're also correct. Very good. The next question is also, I'm assuming no one has a doubt in the last question, right? In case you have, just please stop me or just write it in the chat box. I'll clear this. So, question number 4 is, Johansson proposed pure line theory in which crop? There are 4 options given to you: cereal crop, oilseed crop, conifers, and pulse crop. So, what do you think is the correct answer? D. Yes, Satya, you're correct. The answer is D, because Johansson used Phaseolus vulgaris, right? And they are leguminous plants, and leguminous plants is pulse crops. Yes. So the answer is D. So let's move on to the next question. So a pedigree record of 9214-5-4 F4 designates progeny of plant growing in F3 generation in row number 4, and it was derived from the progeny of a plant growing in F2 generation in row number 5. In which year was the cross made? There are 4 options given to you. What do you think is the correct answer? Uh, why sorry, Satya? Is Oh, no answer. Oh, no, no worries. That's not a problem. You just need to guess. You don't need to be right about it. No worries, okay? So, pedigree method, if I'm correct, it was found by 1927, if I'm right, okay? Just go back and check this. I'm slightly not sure about this. So, uh, if you've seen the uh, video by Sir, you will see that the first 2 letters of a pedigree record gives you the year. Okay, so that means the year ends in 92. Okay, so option B is obviously not correct, and option A is not correct because it is 1492. Pedigree method was shown in 1927 and after that, right? So both of those are not correct, and we haven't reached the year 2092, so that is also not correct. So the correct answer in this case becomes 1992. Was that clear? Does anyone have a doubt in this? All right. All right. So the correct answer in this case is 1992. Just remember that this is the year and this is the number of the plant. Okay, the 14th plant in that row, right? So let's move on to the next question in case you don't have any doubt. So question number 6 is, and this has or can have more than one correct answer. Okay, because it's a MSQ. So the question is, choose the correct option for backcross breeding. Option A is, it is a practice that has been used by breeders for cross pollinating crops. Option B is, backcross breeding is a new technique developed for apomictic plants. Option C is, backcross breeding is repeated until the offspring recovers about 99% genes from recurrent parent. And option D is, backcross breeding is often used to increase the yield of the crop along with the rectification of minor defects. So, what do you think the answer or answers is? A and C? Alright, let's see if that is the correct answer. So, the answer is actually C and D. The reason A is not correct is because it is a practice that has been used by breeders for self-pollinating crops. Okay, because what happens in self-pollinating crops is they keep selfing with themselves, right? So it has the same genotype each time, right? And because of that, you can't have some external trait come into it. Okay, that is not a problem with cross pollinating crops, because if they're of the same species and they cross pollinate, they have a very large variety. You might not need back crossing. Okay, so this is wrong because it's not self pollinating. Okay, so if it was written it's a practice that has been used by breeders for self pollinating crops, this would have been correct. Okay, option B is also not correct because it is not a technique developed for apomictic plants. If you remember, apomictic plants are those which don't require fertilization for forming seeds, like in lemons and orange seeds, right? So they are called apomictic, and since they don't require any form of fertilization, they are not using backcross breeding. Okay, so the correct answers are C and D. C is correct because back crossbreeding is repeated until the offspring that you recover has 99 genes from the recurrent parents. And D is also correct because it is often used to increase the yield of the crop with the rectification of a minor defect. So in the examples that I was giving you, where you were doing either dominant or recessive backcross, you needed only one gene to be transferred to an otherwise perfect plant, right? And that is the minor defect that they're talking about. You only want to transfer one gene. Everything else about this plant is great for you. Only maybe drought resistance or resistance to some disease is what you want to confer into this for it to become better. So the answer to this is C and D. So if you don't have any doubts, I'll move on to the next question. Please stop me or write it in the chat in case you do. Okay. Question number 7 is, progenies derived from single self-pollinated homozygous plant is called multiline, pure line, isogenic line, or inbred. So what do you think the answer is? Option B, pure line. Yes. The correct answer is in fact option B, pure line. So pure lines are progenies derived from single self-pollinated homozygous plants. A multi-line is several pure lines, okay, with difference in just one genes, like some resistant genes which have been produced by backcross. So everything else about them is the same, only that resistance gene is different in them. So they are called multilines. Isogenic lines have the exact same genetic makeup with one gene that is different. Everything else about these plants are the same except one gene. So one gene can be AA or small a, something like that. Okay, everything else about them is the same. Okay, and inbred is cross pollinating species which have been continuously bred with themselves. Okay, they have been selved to get homozygous lines. All right, so it's very rare for you to get homozygous lines when it's a cross pollinating plant. So to get a homozygous line of a cross pollinating plant, you inbreed them. Okay, so the correct answer is B, pure line. If there are no doubts, let's move on to question number 8. Question number 8 is, selection for simply inherited traits starts from which generation? F2, F4, F5, F3. All right, A. Yes, the correct answer is in fact A. And just so we are clear about this, imagine you start off with homozygous lines, homozygous dominant and homozygous recessive. You cross them to get a hybrid, which is F1, which the phenotype will be dominant, and this will be its genotype, right? And you self it, so you get these three varieties, right? That is F2, and these have different phenotypes completely, right? So in F1, if you draw Punnett square, right, you see that all progenies of F1, oh, wait, sorry, all progenies of F1 have the same genotype and phenotype. Only after you self them do you get a variation, right? And so the question was asking you, selection for simply inherited traits starts from which generation? Since these have all the same traits, okay, you can't select from here. You have to start selecting from the F2 generation where you see a difference in phenotypes, right? So the correct answer is F2. You were right. All right. So if there are no doubts in this, I'll move on to the next question. The formation of extreme phenotypes observed in segregating populations compared to phenotypes observed in parental lines is called, there are four options: isogenic line, transgressive segregants, pure line, and heterobeltiosis. What do you think the correct answer is? C. All right. The answer I'll tell you is B. So they are called transgressive segregants. So what happens in this case is, imagine you have AA and, sorry, imagine you have AA and BB and oh, AABB, AABB. Maybe you're crossing these plants, okay? And this particular plant has a yield of 90%, and this particular plant has a yield of 50%, okay? And you get a variety after crossing after F1, F2 may you get a variety AA, BB, okay? In that case, this has a yield of 100%. I'm not doing this very correctly, okay? So I'm just telling you that you had original parents with a yield of 90% and 50%, and after a few generations, you get a plant which has a yield of 100%, okay? So in that case, what you're getting is extreme phenotypes, that is something more than the parental line, and they are called transgressive segregants. So the correct answer in this case is transgressive segregants. Isogenic line, like I told you in a question before, is when you have exact same genetic makeup, only one gene is different. Pure lines are homozygous for only one particular trait. Okay, everything else can be different about them. And the last one, heterobeltiosis, is when a hybrid's performance is better than its parent. So what is the difference between these two? Okay, because in both cases the hybrid's performance, the performance of the offspring is better than the parent. The difference is, in this case it doesn't have to be a hybrid as such. Okay, that is, anything after F2 generation can have anything after F2 generation can have a better performance than the parents, and it's called transgressive segregants. But in heterobeltiosis, the hybrids that are formed in F1 generation, okay, they have a performance better than the parents. All right, so that is the difference. Are there any doubts in this? All right, I don't see anything in the chat, so I'll move on to the next question. This is the last question for today. The question is, which method is mostly used for transfer of cytoplasm? There are four options given to you: mass selection, bulk method, pedigree method, and backcross method. What do you think is the answer? All right. I don't see anything in the chat. The correct answer in this case is backcross method. So in this case, imagine you have one plant from which you want the cytoplasm, because the cytoplasm has your gene of interest. Okay, and you have another variety of the plant from which you want everything else, the rest of the genetic makeup that is found in the nucleus. So what you do is, this becomes your donor plant, okay, and this becomes your recipient plant. Okay, you cross these two, okay, and you get your goal in the end. Okay, whatever plant you're getting, you will keep crossing it with this recurrent parent, and your goal in the end is to have a plant which has the genetic makeup of this, okay, and the cytoplasm of your donor plant. Okay, so like every other backcross, that is what you're hoping for. Okay, so the correct answer in this case is D, backcross method. Satya, yeah, you got it right. Very good. So that is the end of the presentation for today. Uh, is there any doubt regarding any of the questions that I've taken? All right. So if there are no doubts regarding this, anyways, I'll be uploading this onto YouTube. NPTEL team will share the link with you in case you want to go through this again, and they will also be giving you the link for the PPT that I've made, so you guys can have a go at it again. And I'll be having how to join discussion forum. um, Discussion forum, you don't need to do anything. You've joined the course, right? So in the course itself, can you see Q&A in the top, next to announcements about the course Q&A progress? Huh. So that Q&A is the discussion forum. Just try clicking on it, and it should take you to the discussion forum where you can put any questions that you want. All right. Any other doubts? This class will be happening on all Tuesdays, same timings, right? And in case you're not able to join, no worry about it, because there'll be screen recordings for this, and the PPT will also be available to you. If you have any doubts in the assignment questions, we'll be taking them after the due date is over. All right. So if you have no other doubts, you can leave the meeting. That's all for today. Thank you so much for joining. Yes, another doubt, Vivek? Sure. The last date for submitting assignment, if you see the assignments itself, in due date will be written at the top in red. So I think due date for week 1, let's see, just let me check it. Due date for assignment 1 is uh, 5th. I think tomorrow then. If I'm not wrong, it's 5th. And similarly, there'll be in each assignment on the top right corner, you'll see a due date. Can you see it? Yeah, sure. Any other doubts? I can't see anything in the chat. But in case you don't have any doubts, you can leave the meeting. All right. Thank you so much for joining.